Hi.
Okay, please start
Okay.
introducing yourself.
Okay, hi. My name is Selim. I'm gonna be presenting about read mapping. I have this asterisk here because read mapping can be short read mapping or long read mapping, but all my methods are about long read mapping. I didn't want to just change the title after it was decided, so I thought I could just put an asterisk there. Oop. So let's talk about some of the advantages and disadvantages of long read mapping. One main thing is when there are repeating patterns in a genome sequence, short read mapping may be unable to capture it. So long read mapping is obviously better in that. Also, we have what are called structural variants. These can be, what you see when you are calculating the edit distance, such as insertions, deletions, duplications, inversions, and translocations. Just like repeating patterns, if these come in not just one character or two, if there are many characters in your pattern, then you will need long read mapping to capture it. Some disadvantages is the error rate is higher compared to short read mapping. And typically the data that you are dealing with is really big, like hundreds of gigabytes big. So you are gonna have to really be careful about your space complexity and maybe time complexity as well. So this is, it's gonna say candidate mapping, where you can see. Yeah, maybe let's move. Yeah. So this is from Dr. Alsa's review paper. We divide read mapping into two sections. The first one is selecting some candidates, and then there is alignment. Now, in the literature, some people refer to just this candidate selection as mapping. So that terminology can be sometimes confusing, but what I'm referring to is this part plus the next part, alignment. So let's start here. We need to first get some seeds. These are short DNA sequences or words that people refer to in your genome. Once you get your seeds, you want to store them, index them in some data structure, usually hash tables. That's the most common one, right? Also, after you store what you need to get from your seeds, you also extract the seeds from your reads, and you compare what you get from the previous parts, parts B and C. So you have your location list and you have your seeds when you applied part C. So you see which parts are matching. This way you can get an idea of which areas in the genome sequence are good candidates for your mapping. After this is done, there is alignment. And yeah, with the previous 4 parts, these 2 parts, so this in total 6 parts constitute what we call read mapping. So here we have our reference segment on the left. Maybe let me move this again. So we need to change those seeds and filter them before aligning them. And after that, to verify, we look at this edit distance matrix and align them. One thing that when I'm presenting, one thing I like to do is confess what I don't know. So I don't know much detail about this part. So if you have questions, I'm not good at this part. So I have 4 methods that I explored in my review paper.
can bring it maybe.
I mean, right now it's gonna cover like this. Ah, okay. Okay. So I have 4 methods. I'm gonna explain one of them. I'm just gonna mention the other 3 with like 1 sentence. The one I'm gonna talk about in detail is Minimap 2. This is a seed chain extent procedure. So we first extract the minimizer seeds, as summarized, as shown in the top part of the pipeline. Then based on that, we build the hash table index. So it's just like the general pipeline so far. And the next thing is to find some anchors. So this is like the candidate mapping that I was referring to in the beginning. And after we get those anchors, we need to chain them here. So we have the reference and...
How you decide?
I don't know that.
So you collect seed, and what do you do?
So you, for example, look at seed 4. You found some matches, like 3 matches here. So you kind of label them as orange, just like seed 4. And then in each chain, we see some of the labels. Like, and at least from this image, what I got was one label is appearing only once, but I think like that might be just a representation thing. Because if I had more than one appearance of a label, then would that just be the end of the chain? Sure, that could be a method too, but I don't think that's what they do here. And then after that, the last part is quite detailed. We have the candidates that came from the first part. And so based on all the labels, like color coded here, we get the candidate locations. And we extend this kind of result of the chain, the previous step, to get our reads and references.
So why not we just...
So you're saying, like, why not just skip chain 3?
Because just one seed.
So how do you decide that it's noise then? Right? If it's a noise, then by definition, they shouldn't like say it's something remarkable like a chain, right?
But it is a match.
Mm-hmm.
It is something like that match. How we can decide it's a good chain. Random match. We got something in assignment 4.
Yeah.
To process that. What we did.
I can't remember.
There was a threshold.
Yeah.
All right.
Yeah. Yeah, so we decide which chain is the best based on some algorithm like what you mentioned, and then continue with alignment. Yeah. So now in Minimap 2, we need some k-mers, and this is gonna give us like different ranges of k-mers. So from all of the windows of k-mers, we only want to keep the minimum one. Because if you keep the longer ones, that's gonna make the whole process more complicated. I would assume. Huh?
Minimum term quantity. What is minimum?
So I think they are referring to the size
Size of the seed.
of
All
the
them same
window.
size.
Size of the window, right?
All of them same size. We apply the exact same window size over the read or the G. Keep sliding. Within the window, there are multiple patterns. Do something. Then we pick them.
Okay.
Do you remember what is it?
Yeah.
So either we took the lexicographical order, like in English, right? Or we just convert each seed to hash value.
Yeah.
We take the minimum out of those hash value. Value just convert the number.
Okay.
So the minimum.
How are we getting the hash values? Like,
Each
how,
seed
how does the
do
algorithm
mathematical
decide?
equation.
Okay.
Time. You got like 16 character.
Yeah.
So you do something with those 16.
Okay.
So you convert, for example, A into 2 zeros, C into 1, B into 1, 1. This way you already represent all possible characters. And then this becomes a number. A is 0, 0. Right?
Yeah.
Then you do AND operation. There are lots of equations on how you get hash value. We already discussed like sometimes in hash equation or hash value might be good or might be bad based on the collusion rate. So you have a hash table, you want to represent multiple values and to be distinct. So if I have different numbers, different seeds will be represented using the same hash value or totally different. That's what makes one hash equation much better than another equation. Okay?
Yeah.
But we cannot store all the hash values, all the seeds. Otherwise, our table will grow really exponentially. We have to pick some of those seeds. A minimizer was one way to pick those. We don't pick everything, but rather we pick within a window, just give me the minimum seed. The minimum, again, we could define it based on totally different things. One of them is hash value. Another thing, lexicographical order. Something else, as we discussed in the lecture, we look like different approach how we pick these.
Yeah. Okay. Yeah. So, because of the reasons you mentioned, we are able to reduce the space and the time that it takes for us to look up what we need. So we get the positions of the minimizers as well. We need to store those to later use as our reference. And because we have those stored, we can just instantly look them up. So this is gonna save a lot of time. Okay. So when we move on to the chaining, we look at our anchors and we look at which of the anchors are matching the minimizers from the previous step. And then we score them. Again, here there are some different algorithms for scoring. Not everybody does the same thing. If I remember correctly, like there were some papers just based on how they do the scoring. Like they said, oh, okay, this is a paper-worthy new thing just because we changed how we score. And the next part is we want to avoid taking too many seeds. For this, dynamic programming is used. Then the last part, that E and F part from the main pipeline, alignment. Now, out of the chains from the previous step, again, we filter through them and we select the top ones. And this is a common method, Smith-Waterman, that helps us compute a detailed alignment.
What does bandit?
I just copied it from the paper, but I don't remember right now. I think it was about like kind of like the length of the. It was somehow related to the k-mer, the k value. I don't know what exactly it was doing, but as the k was increasing, it was proportional, something like that.
Does anyone? Number of diagonals, not columns, not
Number of
just wanna
diagonals.
reduce the-
Okay.
Get the main, then
Uh-huh.
we count how many to the right, how many diagonals. So if we say
You
back
mean like in
forward,
the edit distance calculation?
it's related to edit distance,
Uh-huh.
but not directly equivalent to the same.
Okay.
It can dictate maximum edit distance calculate, but it is related to the distance line or the edit distance algorithm. The way
Okay.
to optimize calculation, instead of calculating the entire dynamic programming table, we just calculate a portion scattered around the main diagram.
Okay. So when you say like it drops some diagonals, like you mean you have your whole table. Let's say you drop some diagonals on the top right, or is it something like that? Okay. So if you're removing just two diagonals, let's say you remove two from here, the middle stays always. Or,
It's rather we focus on the main diagonal and we how many we consider rather than deleting. How many we keep two to the right, two to the left of the
oh, that's where bandit
everything
comes
else
from.
is just
Okay.
band
Okay.
because it's just band, right?
Yeah.
It's like a river and fixed sides all over the table.
Are you always keeping the diagonals adjacent to the center diagonal? You're never skipping, right?
Because at each cell we have to calculate 3 cells in it.
Yeah.
So everything should be computed in order to calculate that.
Okay. And yeah, we get our detailed alignment, and then we get what are called base level alignments. And again, because we were able to chain, we made our algorithm more efficient this way, especially for long reads. So for, yeah, again, all these algorithms are gonna have the same advantage because they are designed for long reads. So I guess it's gonna be yeah, in every algorithm's advantage, very fast, efficient for huge genomes. These are designed for those, so it's naturally there. And Minimap2 specifically can handle both DNA and RNA. Now, we will not see this in every algorithm. And another advantage is that the are these two parameters. The disadvantages can be very, it can have some misalignments when you're looking at those repetitive patterns for what are called low identity regions. And sometimes the small variants are also missed. And these are the three other methods that are used. There's BWA MEM2, which is very good at handling multi-core CPUs. And there's WinowMap2. This one uses minimal confidently alignable substrings. to improve mapping in highly repetitive regions. And there is parameterized sync-merge schemes. This one is not a mapping algorithm itself. This is a seeding method that's built on top of Minimap2. So basically I have 3 new mapping methods and 1 seeding method. And here I have 2 tables. The first one is the results from the literature, the 4 methods as they are reported in their papers. And the bottom table is the results that I got when I ran the algorithms. The data set I used was smaller than what's used in the papers. And here this column is empty because getting those repeats is a non-trivial process. It's kind of complex. I didn't.
What do you mean by repeat?
So they first do their mapping normally, and then they decide like they get the repeating patterns based on that mapping, and then they kind of run it again. Like some of the algorithms, I don't know if all of them are recursive when they are doing this. So you need to be able to run it recursively, and that seemed kind of difficult to do for some of them. So I just skipped that one.
So how do you evaluate?
I have the other metrics that they use, the indexing time, memory that's used, and the runtime.
Oh, so is this done by another paper or yours?
These are my results. So the first one here, Minimap2, the same method on a different data set.
Yeah, but this number is yours or?
These are my numbers. Yeah. These are from the literature.
I see.
Yeah. First table from the literature, second table, my
But
own.
not the same data set.
No.
Not the same FASTQ, not the same.
No.
You build tracks in advance or?
So this is what I found online, one of those websites that you share. And I got a subset of 10K reads from that to be able to do it quickly. And then I ran the methods on them as they are described in the GitHub pages of the methods. Now, one thing I want to mention is this one. I don't have an indexing time from Map2 because it didn't have like an indexing command on their GitHub page. So I'm assuming for the paper itself, they they knew where the algorithm for indexing time was in their code, but I looked at their code. I wasn't sure about the.
So it's not mentioned.
No. For the other methods, they just have an indexing command, so it's super easy.
I thought when you map to
What PSS is built on top of Minimap.
No,
Winomap.
That same source code, we change in the code itself, in the algorithm of Minimap. So probably it's exactly the same.
Yeah, it's probably there, but I wasn't sure like
Okay.
where it started, where it ended, so I didn't want
based
to
on
report
those
something
results,
accurate.
algorithm,
So to look at that, I'm mainly looking at these 2 columns. So I can comment on the indexing time and the memory. And let's look at indexing time. So the best one seems to be Minimap2 and PSS, which is again Minimap2. And in terms of memory, it's Winomap2. That's gonna require less memory. But the problem with Winomap2 is even in that subset that was not giving me any unmapped reads, in the other methods, it's giving over 1% on unmapped reads.
Yeah, that's excellent. So as you can see from the numbers, you cannot tell what is best based on the execution time only or the indexing requirement for memory. So you have to look at all these metrics. For example, unmapped read. If the tool cannot map most of the read, or there is a significant fraction of the reads cannot be mapped, this is bad, even if the tool is very fast. The reason, if you don't map a single read, then you are missing something of the information. Coverage of the read.
Yeah. And that's all I have for the presentation.
Great. We have about 3 minutes for questions. Yeah, please ask questions. One strong question. And grill him. It's okay.
Yeah, good.
Yeah, go ahead.
Yeah, this is bigger than what I had. I mean, if I had the patience to just wait 7 minutes, I guess it would be fine. But yeah, I thought they already used that data set. Sure, it would be better to double check that what they did was right. But I kind of blindly trusted them and ran it on a different data set to see if it actually works. Yeah. I hope you guys didn't. I hope you also tested your own literature reviews data sets. No?
Yeah, please make sure you add all command lines.
Okay.
Make sure everything, including the data set you use, exact command line used to run, and so on. And this is the same for everyone. Right? I want to see this in the report. And if you use some Conda lines to run or install a tool. So whatever you use for the experimental result, any instruction, think about you send this to your colleague and you ask them to reproduce these results. Any instruction would be helpful, please include it. If you download the tool from somewhere, if you install it, if you run it, if you did some third-party evaluation or benchmarking tool to generate those results, because what you get from those tools is just SAM file, right? So you need another tool to take those 4 SAM files and compare them together. So if there's a script or there's some extra tool that you use to evaluate or compare those SAM tools or SAM files, please include that as well. Anything related to the experimental result, please include it. All right? Any more questions? We have 1 minute.
The TA doesn't respond to emails. Maybe it's just me, I don't know. I was asking if there's going to be a submission box.
Yeah, please try to CC me, all correspondence. See me. For the submission, we'll give you more instruction. As I said, I'll give you a few days. Try to fix this comment in your report, then you can send it. Within 2 days, hopefully, we'll have something on my college. But I would say the grade that you'll get right away. Once you present, you'll get something out of it. Based on everyone evaluation, not based on my evaluation. So maybe we can give it 1 minute to submit your evaluation. And then the next presenter maybe can send the slides to my email right away. Thank you so much, Selim.
You're welcome. Thank you for listening.